Hi, so this is going to replace our second lab that we're going to do in Biology 60. I have made this document available to you on Moodle, but I'm going to redo these videos and replace them with this one right here. So this is me, this is our lab, and this should give you a better idea of what's happening in this lab and give you the chance to do it independently if you aren't able to come in. So lab 2 is on osmosis and diffusion, and basically we're going to set up a mock up of a cell. And so I have a beaker here of distilled water, and I also have some dialysis tubing. So that's just a cellulose tube, kind of like a sausage casing, that's been filled with a starch and glucose solution. It has a little knot in the end and a clip on the top. I'm basically going to go through the results of this lab in this video. And so what you're missing out on is kind of the, time it takes for this to come to equilibrium. I also have an image up on the next document I'll share with you so that you can kind of see what's going on. So to get started, I'm going to recreate these initial results. And so you'll be using the same handout as we are in class, and any of these ones with asterisks were results that we recorded ahead of the lab. And so basically what I'm saying is, if I test the dialysis tubing before, I should find that there is starch and glucose in there. And so we want to know what a positive test for starch and glucose actually is. I'm also going to take and test the distilled water that the casing is going to be in to see if there is starch or glucose in the distilled water. So like I say, most of this is just so you can have an idea at home what we're doing. To test for the dialysis tubing before, I'm going to take some of this sample and I'm basically going to see what a positive result for glucose and starch looks like. And so if I take a little sample, I think I have one right here, then I will take my urinalysis stick. So this is to test for glucose, and it's often to test for glucose in urine, which would be a sign of diabetes. If I dip it in there and let it sit for about 15 seconds or so, try and keep an eye on the time, it should turn to a color indicating that there is glucose in the dialysis tube. So I don't know if you can see the color, from nothing up to some. And so I'm kind of midway on that scale, actually moving up to a little more glucose. So these are just qualitative results. And in the dialysis tubing, yes, there is glucose before. Let's take that same little test tube that I have that has the solution that's going to go in the dialysis tubing, and I'm going to put some Lugol's solution in there. Now, Lugol's solution is just an iodine-based solution, and in the bottle, it's sort of this brown color, sort of amber almost, but fairly opaque. And when I drop it in, a positive test for starch is that it goes kind of an inky blue-black. And so I have the test tube here. I'm putting this brown liquid in, the Lugol's in, and it should go inky blue-black. So that one, oh yeah, there it goes. So it's got a very black color to it now. It's actually got almost a bit of a greenish tinge to it. And so that's a positive test for the Lugol solution. So the results here is that, yes, there is starch in the dialysis tube before. So you remember that solution is in my little sausage casing here. And what I'm going to do is take the mass of this as well, and that's also recorded on my chart. And I'm also going to test to see if there is starch or glucose in distilled water. Now, you could probably get ahead of me on this one. So this is a little test tube of distilled water. And if I dip it with the urinalysis stick, there is no sugar in water. We can see here that this is a negative result. So there's nothing in that as far as glucose goes. If I were to take that same little test tube that has the distilled water in it and drop it with the Lugol's solution, what I'll end up doing is not having that changing color, that inky blue color, greeny color. It'll just dilute and be more of that amber yellow color. And so if I do that, we can see that it's much different. It's very just yellow or amber color, right? So this is a negative test for starch. That means that there's no starch in distilled water. So these no-nos down here, we fill out in the lab ahead of time. So you're going to have something set up that looks a lot like this. So we'd have our dialysis tubing at the beginning in my beaker. And this image is great because it kind of shows the dissolved things, what's where. And we're going to let this sit for a half hour. In the lab, we're going to do the second part of the lab while this is going on. You'll be able to fast forward because you don't have to wait. I'm going to show you how it goes anyway. So imagine half hour has passed. After, when things have reached equilibrium, this diagram over here is illustrating that. And so this could be a handy one if you freeze frame. You can kind of see the dissolved things. If I switch back to my results, I've pre-recorded these for us. And so what I'm going to do after a half hour is cut open the dialysis tubing bag and test the solution that's in it, and also take and test the liquid that that dialysis bag has been soaking in. And so a few key things are going to happen, and they're recorded here on the chart. So these are after results. You'll notice that there is a change in the mass of the dialysis tubing, and you're going to have to theorize as to why that is in your lab results. The dialysis tubing bag still has starch in it, and it still has some glucose in it. Okay, after. The beaker that it was soaking in, if I test that with the glucose test strips and the Lugol's solution, I find that there is no starch in that water, even though there was starch in the bag. And there is now glucose in the bag. So the key thing with this is that the dialysis bag is not selectively permeable like a plasma membrane, but semi-permeable. So it sorts based on size of molecules. And now that we've studied those macromolecules and some of the chemistry, you should have an idea of how these things sort out. So it sort of allows things that are smaller than the pores to pass and blocks anything that's larger than the pore from passing through, even if there is a concentration gradient. So your lab analysis asks you to use all the words: concentration gradient, diffusion, osmosis, maybe some tonicity in this lab to explain these results. So those are your results, and that should hopefully give you the idea of how the lab goes as a somewhat replacement to being here. The other thing that I'd mention is that on the lab, oops, sorry, wrong document, the part B, for the second part of the lab, you can use these images. So these would be onion cells. I have a red onion here in the lab that we cut up and look at the onion cells, the same cells in distilled water and then in salt water. And again, that part, you're asked to use all the vocabulary of cell transport to explain what's going on. All right, good luck. And if you have any questions, let me know.